Stanford University.
Thank you very much, Rob, for this introduction. I had no idea who I was. I'm not sure that that's me, but anyway, I'll hire you. I can't pay you, though. So for me, this is a particular honor to be here. It's very special for me to give this talk. We all give a lot of talks, and but you know, most talks are kind of routine, and this is special because this is the inaugural symposium for the Stanford Neuroscience Institute, as you all know, and it's really an effort to do something new at Stanford, to try to unite the often disparate and not always friendly provinces of neuroscience into one big community, which I think is terrific. And I really thank Bill for taking such a serious stab at it and trying to get this done, which I think is absolutely admirable. What I want to do today is talk to you about Neurexins and convince you that they're worth talking about. And my talk has three parts. I'll first start off with some very general considerations about neural circuits. I will then try to tell you why Neurexins might be important. And I will finally add some detail and talk about two brief stories on Neurexins. So let me start with the first part. You all know, agree, I hope, that the best way where we can all talk about how the brain physically mediates, how the brain physically functions, what mediates brain function, is to focus on neural circuits as sort of the element that everybody can agree on is a way to understanding the brain. And what you see here schematically is just a microcircuit, if you want, a feed-forward inhibition circuit, where when you excite this neuron, it excites the next neuron, but it also excites an interneuron, which then inhibits this neuron. And depending on the exact firing pattern of this neuron, there will be an input-output relation that also then depends on the activity of this neuron. The standard way that has evolved recently in analyzing the relationship of neural circuits to behavior in particular consists of three components. One is to map the connections that make up a circuit, using, for example, viral tracers or color toxin. One is to determine which neurons and connections participate in a particular behavior, as, for example, beautifully described by Carl this morning using optogenetics. And the third one is to actually measure spike patterns of neurons during a behavior, for example, using in vivo recordings, which are crucial for trying to understand which neurons are activated in what pattern during a behavior. And I think these types of approaches that are usually mixed two or three together are really valuable in trying to understand how particular circuits relate to particular behaviors. However, there's also a few issues here, okay? And so one is just the sheer scale of the brain. There's so many synapses, there's so many circuits, there's so many behaviors. At this point, most people pick their favorite brain area and their favorite behaviors, usually behaviors that we can relate to, you know, like appetizing behavior, what we like, or addiction, or things like this. But there is a lot of stuff there. And Carl, again, elegantly alluded to the fact that just collecting enough data about circuitry, just pure data, is a computational challenge if we could get enough of it in the first place at the level that we needed. The other issue that we need to remember is that these analyses do not explain a behavior. They shed light, they correlate on what kinds of activities are involved in a behavior. But the fact that the behavior changes when a neuron fires does not mean that the neuron's circuit actually normally mediates the corresponding behavior. And so obviously we all want to understand better how this works, and it's going to take a while. But what I would like to argue that is, in addition to these very valuable approaches, we need a complementary insights into the general principles that apply to all neural circuits. in some way sort of be able to reconstruct circuits from first principles. And obviously, these types of insights needs to be molecular. And so to start off with this, what are the first principles of circuits? It's obviously that circuits are formed by synapses. And synapses transfer information from one neuron to the next, and they process information. They have specific properties. They change as a function of use. And 3 parameters about synapses are particularly noteworthy. First, synapses are formed at specific sites, which is important. They are formed at specific sites not only defined by the identity of the pre- and postsynaptic neuron, but also where on these neurons these synapses form has an enormous importance for understanding how the circuit in the end operates. Second, synapse properties differ depending on the pre- and postsynaptic neuron. So even the presynaptic properties of synapses formed by this neuron differ depending on whether the postsynaptic cell is another pyramidal cell or an interneuron. Among these parameters that differ are release probability, kinetics of release, postsynaptic receptor composition, plasticity, etc., etc. And finally, possibly most importantly, synapses are plastic. They are plastic in the short-term time frame, they're plastic in the long-term time frame, they're plastic in many different ways. What you see here is just one illustration, which was taken from a paper by Wade Regehr, illustrating that the same postsynaptic Purkinje cell neuron in the cerebellum exhibits different synaptic short-term plasticity depending on which input fiber is here. And if you translate this or sort of transpose this on here, it's easy to imagine that having facilitating or depressing synapses has an enormous effect on the input-output relationship of the circuit. So the spike patterns of a neuron are themselves not informative enough to tell us what the neuron actually does if we do not know how such patterns are translated into postsynaptic signals at its various efferent synapses. So what I hope these considerations illustrate is that synaptic parameters dictate the input-output properties of neural circuits. And so how does this relate to the role of circuits in behavior? What I want to tell you now is one particular experiment that was actually performed by Wei Shu, who is postdoc in our lab, who's sitting, I think, right there. And that initially was inspired by yet another experiment that he had performed earlier. And this experiment was to inject into the prefrontal cortex of mice AAVs that inactivate neurotransmitter release, and then to measure in these mice the effect of the inactivation of the prefrontal output on fear conditioning memory. Fear conditioning memory is measured by basically putting a mouse into a defined chamber and then pairing a cue, a tone, with an electric shock, which the mouse doesn't like, not surprisingly. And then the mouse remembers the context as well as the cue. And traditionally, what people do is they measure how frozen the mouse becomes when it gets back into the same environment because it expects another shock, or when it hears the auditory cue. And these are called contextual and cued fear conditioning. What Wei did, he introduced yet another parameter here in that he also measured the freezing of the mouse without the cue in an altered context to assess the precision of the fear memory. And what you see here, down here, are the results, which were quite remarkable because the prefrontal cortex had previously been shown not to be involved in either contextual or cued fear conditioning. And indeed, in the training context, there was no difference between control injections and 2 different types of inactivating outputs, synaptic outputs from the cortex. However, the precision of fear memory was severely impaired, as you can see here. The mouse froze more in the altered context than the control, which was less than it was in the real context, as you would expect for the control, demonstrating that the mouse didn't quite remember anymore what the real context was. And again, cued fear conditioning was the same as expected from the literature. So these experiments prompted us to search for a synaptic output pathway from the prefrontal cortex that would be responsible for this unexpected inability to remember exactly the condition under which the noxious stimuli occurred. And so what Wei did is he developed a way of tracing outputs, which we call Synaptotag, from the prefrontal cortex, and he identified a major projection into a completely obscure nucleus in the midbrain, which is called the nucleus reuniens, and which I actually had never heard about, and I'm sure most of you haven't heard about either. But anyway, now you will remember. Hopefully, maybe not. Depends on the noxious stimuli I give you. Anyway, so what he did then is he inactivated neurons in this nucleus, again, using tetanus toxin to inactivate the efferent outputs. But he also, in a separate experiment, activated these neurons by knocking down an inhibitory cell adhesion molecule, thereby making these neurons more active. And then he looked at the effect of these manipulations on the fear conditioning. And what he found was that contextual fear conditioning was, again, perfectly normal, but the altered context was changed in such a way that inhibiting the output made the mouse freeze more. So it thought that the altered context was the real context, whereas activating the nucleus reunions achieved the opposite. The mouse now thought that it was that there was no resemblance at all. So there was a bidirectional control by these neurons. Acute fear condition was the same, and a discrimination index just indicates how well this mouse discriminated between the real and the altered context. And so in other words, what these experiments demonstrated is that the activity of these neurons bidirectionally controlled memory precision, but not the memory itself. And this raised the question whether this is really a function of the firing code, of the firing activity, or whether this is a function of whether or not the neurons are active at all. And so in a final experiment, what Wei did is use the brilliant invention of optogenetics, and he introduced a channel adoption into nucleus reunions, and then he used two different stimulus paradigms during the training, fear conditioning training, either tonic stimulation or pulse stimulation, after which he measured, again, the same three fear conditioning paradigms. And what he found was, as you expect, there was no effect of either stimulation on contextual fear conditioning. But if you look at the altered context, or better, even the discrimination index, there was a dramatic difference depending on the stimulation frequency, in that phasic stimulation basically made the mouse forget that this context was similar, and tonic stimulation caused the opposite. The mouse now thought it was in the context it was shocked in, whereas it wasn't. And finally, the tone is the same. And so the reason I am telling you all this is that this experiment to us illustrates that it's really the pattern of excitation, in this case of these neurons, that encodes the memory precision. It's not just broadly activity, but it's the specific spike pattern. And the consequences in terms of looking at the circuit logic, at how circuits operate, is that we can't just look at circuits as such in terms of connectivity. We can't just look at what synapses are formed and how neurons fire. We actually need to be able to reconstruct how these synapses operate at any given position. And so this makes these various properties of synapses incredibly important in order to understand the overall system. So given the challenge of describing the brain's trillions of synapses and millions of parallel and overlapping circuits, a complementary approach may help in this case, which is to decipher what I call the molecular logic that builds synapses and circuits and will allow prediction of their properties if we understand that molecular logic, which is where Neurexins come in. So we believe, and I hope when you walk out here, you believe too, that Neurexins are sort of the linchpins, are key organizers for the molecular logic of neural circuits. And so what I want to do is briefly introduce to you what are neurexins before I tell you two more detailed stories that sort of shed light on particular facets. It all started with this critter, which is a black widow spider, if you don't know what it is. Known obviously for its graceful romantic entanglements. Graceful from the female side, I suppose. But it has one, for us, even more interesting property, which is that it makes a toxin, which is called latotoxin. And this toxin binds to presynaptic terminals and causes neurotransmitter release. And so a long time ago, more than 20 years ago, we set out to purify the receptor for this toxin as an avenue to understanding how neurotransmitter release works. Now, it didn't tell us how neurotransmitter release works, but it did open up, in many ways, a new perspective on how synapses might be organized. Because from the very initial cloning of neurexins, it became apparent that these molecules look like cell surface receptors that are highly polymorphic due to extensive alternative splicing. And so we postulated that there might be cell recognition molecules in the nerve terminal that might instruct the nerve terminal how to behave. And this prediction is now slowly, after more than 20 years, coming to be validated. Neurexins are cell surface proteins. They come in two flavors, alpha and beta neurexins. The beta neurexins are shorter than the alpha neurexins. They're basically N-terminally truncated alpha neurexins. They have this multi-domain structure. On the extracellular side, a short intracellular sequence. There's 3 genes, and each gene has an independent promoter for alpha neurexins and for beta neurexins. So there's 6 principal forms. The neurexins have very extensive alternative splicing at 6 sites, creating thousands of isoforms. They bind to intracellular PDZ domain proteins such as Cas, and there's many gene mutations associated with autism and schizophrenia in their genes. The baroque alternative splicing at 6 sites is what initially caught our attention. And most of this alternative splicing appears to be highly regulated, as I will show you on the example of splice site number 4, which affects both alpha and beta neurexin in this LNS domain shown here. And what this slide illustrates as a way to demonstrate to you the regulation of this alternative splicing, on the top you see the total levels of these 3 neurexins, 1, 2, and 3, in these various broadly dissected brain regions from mouse. And below, you see a ratio of the 2 splice variants at this one particular site, namely plus, which is splice site in, minus is splice site out. And you can see already from this pattern that there is a segregation between brain regions in terms of what the alternative splicing looks like. Just to illustrate this further, in the cortex, for example, it's primarily splice site 4 out, with neurexin 3 having the biggest effect, whereas in the adjacent striatum, it's more primarily splice site 4 in. And since this is a logarithmic scale, this is a 100-fold difference, so the effect is quite significant. What does this mean, and what do neurexins actually mean? One of the issues that came up very early is whether neurexins are, in fact, synaptic. So we tested this using immunocytochemistry and using a Knockin mouse that introduces an epitope. And what we found is that they are indeed presynaptically localized by this criterion. What you see here is a labeling of these synapses containing neurexins, endogenous neurexins, either co-stained with a presynaptic marker, and you can see perfect overlap, or with a postsynaptic marker, and you can see adjacent properties. So with these properties of being both localized to synapses and highly alternatively spliced, the next issue that came up is whether they're important at all. And again, some time ago, we asked this question initially using a constitutive knockout approach, because about 15 years ago or so, that was the only approach that was available. And so we made constitutive knockout mice for all alpha neurexins. And we found that the alpha neurexins had a severe phenotype. The single neurexin knockouts were viable but behavioral abnormal. the double Neurexin knockouts largely died, and the triple Neurexin knockouts were completely lethal. Now, you have to understand that for a postdoc in our labs, this is a wonderful result, even though for the mouse it is not. Because there's so many knockouts we have made that don't have an apparent survival phenotype. At least this tells you that it's important. The mouse needs it. And so we wondered what's happening in these mice. We found that there was no loss of excitatory synapses. There was no apparent change in neuronal development, but there was a severe impairment of all synaptic transmission. And I'm not going to go into detail. I just want to show one slide from this study demonstrating that using this stimulation paradigm as shown here, and here the postsynaptic responses measured electrophysiologically in the single knockout and the triple knockout. And you can see that in comparing the triple knockout to the single knockout, there's a massive decrease in amplitude. There's very little left. And there's a massive increase in the so-called failure rate, which measures how often there is absolutely no response at all. In other words, what these mice thus suffered from was a fundamental loss of nearly all synaptic transmission, demonstrating that the Neurexins are essential for synaptic transmission as such. And in many ways, that was a valuable result because it confirmed the importance, but at the same time, it was a dead end. Because if nothing works, you can't really figure out what a protein does because it could do everything. And especially with constitutive knockout mice, it was impossible to actually dissect this. So what I'm going to, so these results thus demonstrated that they're fundamentally essential, and they demonstrated that they must be doing something important, but they didn't actually tell us exactly what and how. So in subsequent years, we went ahead and tried to analyze this further and to get further insight into this question. And one issue that obviously came up almost immediately from the cloning of the Neurexins is that they must interact with postsynaptic ligands because they look like presynaptic receptors, so there must be something there postsynaptically. And over the years, our lab and many other labs over the world have identified a series of ligands that by now I think are number close to 10 different ligands, 4 of which are shown here. And these 4 ligands share the fact that they all bind to both alpha and beta Neurexins with nanomolar affinity. And just to illustrate this diversity, neuroligands were identified first. They are highly evolutionarily conserved. Cerebellins were recently identified by Machina's lab. They're soluble proteins that bind simultaneously to postsynaptic receptors and presynaptic Neurexins. LRTMs are also cell surface proteins that we, together with Rob Malenka, identified as Neurexins ligands some years ago, and serolatophilins are GPCRs. So there's very different structures here that all are nanoaffinity ligands for Neurexins. They have nothing in common except for the fact that they bind to Neurexins. Remarkably, some of these ligands are actually regulated by alternative splicing, and specifically, these particular ligands, as it turns out, they're all regulated in their binding by alternative splicing of Neurexins to splice site number 4. In that, when you have a splice site 4 insert here, cerebellins bind. But if you have none, cerebellins don't bind. Neuroligands bind to plus minus, LRTMs and serolatophilins only bind to minus. So what the picture that emerges from these studies is that of a dynamic interaction network that switches with small shifts in gene expression or alternative splicing. Finally, mutations, as I already alluded to, in genes encoding Neurexins and their ligands are linked to autism and schizophrenia. Neurexins are generally linked. There's been hundreds of mutations. There's been many mutations in neuroligands. LRTMs are linked to schizophrenia. Shanks are likely to the most abundant, the most frequently mutated gene in idiopathic autism. CASC has been linked to many different neuropsychiatric disorders, MINTs, and others. And in fact, Neurexin 1 is probably still the most frequent single gene schizophrenia mutation as far as we can tell. So I hope that this sort of blitz through the Neurexin landscape, if you want to call it that, convinces you that these are very important molecules for understanding how synapses might work. And what I want to do for the rest of the talk is very briefly tell you about 2 facets that illustrate the diversity of Neurexin functions. And one
relates to the beta-neurexins that I already mentioned earlier as being the truncated variants of alpha-neurexins that are produced by all neurexin genes. And the overall question here was, why do all neurexin genes produce beta-neurexins if the alpha-neurexins already do so much, as I showed you a few minutes ago? And so we generated knockout mice, conditional knockout mice, triple knockout mice, and analyzed them in the hippocampus. And these experiments were largely performed by Garrett Anderson recently in the lab. And the way how he did this is he, again, performed AAV injections into the CA1 region of the hippocampus. And then a couple of weeks later, he cut slices, and he measured electrophysiologically the postsynaptic responses on subiculum neuron upon stimulating presynaptic axons arising from these infected CA1 neurons. Now, the subiculum contains two predominant types of neurons, which makes things a little complicated in that one type of neuron, which is called the regular firing neuron, fires regularly. And the second type of neuron, which is called the burst firing neuron, fires surprisingly bursts. Okay? So these two types of neurons have been distinguished by the firing properties. And interestingly, these two types of neurons also differ from each other because they have completely different types of LTP, long-term potentiation, a long-term form of plasticity, in that the regular firing neurons have an MDA receptor-dependent postsynaptic LTP that Rob has characterized so well over the years, whereas the burst firing neuron has a presynaptic time of LTP. So they're quite different in many ways. And so we asked, what is the effect of deleting beta-neurexins on presynaptically on postsynaptic responses? And what we found using input-output curves where we increase the stimulus intensity and measure the size of the postsynaptic response, that in one type of neuron, the regular type of firing type of neuron, there was very little of an effect, maybe a trend for a decrease. But in the other type of neuron, there was a massive decrease in the strength of synaptic transmission. And experiments that I don't have time to show you today demonstrated that this may be associated with a decrease in release probability, which in turn was associated with a decrease in the amount of calcium that flowed into the nerve terminal during an action potential. And so these properties are signatures of the effects that a class of molecules called endocannabinoids are responsible for. Endocannabinoids are agents that regulate synaptic strength, and that based on these findings became candidates for these effects. Just to illustrate what endocannabinoids do, they're diffusible signaling molecules just like endogenous marijuana. Basically, marijuana, if you smoke marijuana, I'm sure nobody here ever has smoked marijuana, but anyway. If you do smoke marijuana, that's what it mimics, yeah. They're produced postsynaptically, they diffuse across the synaptic cleft, and they bind to presynaptic CB1 receptors and inhibit neurotransmitter release. And so to test whether subiculum synapses are normally regulated, we first had to test whether subiculum synapses are normally regulated by endocannabinoids. And so we added endocannabinoid, an inhibitor of the CB1 receptors, to the synapses during stimulation, either the regular firing in blue or the burst firing in red. And you can see that the regular firing in blue has no change, but the burst firing in red gets disinhibited. It gets bigger, and this is summarized here, demonstrating that only one of the two types of cells, in fact, the burst firing cell, the same type that was affected by the beta-neurexin knockout, is actually disinhibited. So this led us to ask, what is the relation to the beta-neurexin knockout phenotype? And we found that indeed, when you look at the wild-type control here in gray, and you knock out the neurexin, the disinhibition becomes bigger, actually significantly bigger, demonstrating that at least in part, the impairment in neurotransmitter release during the beta-neurexin knockout was due to increased endocannabinoid signaling. And so this was kind of surprising. And it led us to ask, since there is this difference between burst and regular firing neurons in LTP, whether there's also a relationship here to LTP. And so, as you may recall, there's these two different types of LTP in the burst and regular firing cells. So we asked whether they are affected by endocannabinoids in these mice. And what you see here is that in the regular firing subiculum neurons, there's no effect whatsoever. There's very robust LTP, 200%, no change with the beta-neurexin knockouts. But when you knock out beta-neurexins, LTP is basically gone. It's almost completely inhibited, demonstrating that this signaling, endocannabinoid signaling, may not only affect the release properties, but also the ability of the synapses to be potentiated presynaptically by this presynaptic form of LTP by the knockout. And so we ask whether this is really true by basically adding back an AM255, a endocannabinoid receptor inhibitor, and you can see that in the wild type it had no effect on LTP, but in the knockout it completely rescued LTP, demonstrating that it was indeed an increased endocannabinoid tone that retrogradely regulated this. And so to summarize these studies, what these basically showed is beta-neurexins affect endocannabinoid production such that in the absence of beta-neurexins, endocannabinoid production goes up, activates CB1 receptors, and that decreases release in LTP. And this type of regulation has obvious consequences for circuits, for understanding how circuits work. And one way to test this is to go back to what I told you before, to go back to the fear conditioning paradigm, use the same injected mice into the CA1 region, but now measure their ability to remember context in the fear conditioning paradigm. And what you can see, if we inactivate the beta-neurexins here, there's a loss of freezing or fear conditioning in the contextual memory, but the precision of the memory and the acute conditioning remains the same, demonstrating that presynaptic beta-neurexin knockout impairs contextual fear conditioning. In other words, that this type of regulation that I demonstrated to you, that depends on beta-neurexins, is essential for normal formation of contextual spatial memories in the hippocampus. So the implications are that beta-neurexins perform non-redundant functions not mediated by the more abundant alpha-neurexins, which I find surprising because they are right there at the same time. That presynaptic beta-neurexins regulate postsynaptic endocannabinoid synthesis and/or secretion, which is a form of transsynaptic synapse specification, and that postsynaptic endocannabinoids control presynaptic LTP in a manner dependent on beta-neurexins. Okay, in my final few minutes, I would like to talk about a completely different story that in some ways has some similarities, but in other ways also illustrates to you how different everything can be just by switching to a different component, a different aspect, facet of neurexins. And here, the overall question is not what alpha or beta neurexins do, but what is the physiological significance of this regulated alternative splicing at splice site number 4? And remember, this regulated alternative splicing is not only regulated, it also regulates all these interactions. And so we wondered, does it have any physiological significance? And in order to ask this question, we generated mice that change alternative splicing in a manner that we can manipulate. So we generated mice in which alternative splicing was changed in such a manner that it became non-alternative, it became constitutive. We caused point mutations into the gene that rendered the alternatively spliced sequence always included. In addition, we made the alternative splicing constitutively excluded using Cre recombinase, which excises the alternatively spliced sequence. So this Knockin causes constitutive inclusion of splice site number 4, whereas Cre recombinase causes constitutive excision. And so this allows us, in one neurexin, neurexin 3, to manipulate at will in a mouse what happens to this site of alternative splicing. Does this have any effect on synaptic transmission? It does, and it has a major effect, but it has a surprising effect. It selectively impaired synaptic responses that are mediated by one type of glutamate receptors at excitatory synapses. What you see here is an amplitude plot of the synaptic responses. You can see that the wild type and SS4 minus condition are the same, but the SS4 plus constitutively included is almost halved. However, if you measure the same synapses, but now the response is mediated by a different postsynaptic glutamate receptors, NMDA receptors, they're the same, as are inhibitory responses. Moreover, this phenotype was rescued by all Neurexins containing SS4 minus splice sites, but by none containing SS4 plus splice sites. So this was in many ways surprising because it suggested that the presynaptic manipulation of these neurexin alternative splicing led to a postsynaptic selective loss of AMPA-Receptors. And indeed, using immunocytochemistry, as shown here in these pictures and quantified down here, we found that the density of synapses was the same, but that whenever there was SS4+, as shown here for two different types of AMPA-Receptors, the amount of AMPA-Receptors postsynaptically was approximately halved per synapse. And this demonstrated that there was a loss of postsynaptic AMPA-Receptors that was mediated by the presynaptic deletion of, by the presynaptic manipulation of splice site number 4 in Neurexin 3. And this loss, other experiments showed, was due to increased AMPA-receptor endocytosis. So it was basically removed from the synapses. So these experiments really raised question whether Neurexins really are presynaptic, because this is a purely postsynaptic effect, and they also raised the question how this might work. And so to address this, we used again the same preparation that I showed you earlier, where you have these two different types of neurons. You inject your viruses into CA1, and you measure in the subiculum to really specifically measure only presynaptic effects. And what we found, different from the beta-neurexin manipulation, that in this case, in both types of neurons, as shown here, synaptic strength was dramatically impaired when you had an SS4+, which is the control condition for the Knockin mouse, but not when you have an SS4-, which is the precondition, which is the same as the wild type, as shown here. So both types of synapses, both types of neurons were in vivo impaired by presynaptic knockout of presynaptic manipulation of alternative splicing at splice site 4 of Neurexin 3. So this basically confirms that this is physiologically happens actually in a real animal, and that you have a presynaptic effect on a postsynaptic AMPA-receptor response. What about LTP? And remember, subiculum neurons exhibit these two different types of LTP. So we tested them again separately, and we found now that the other type of LTP, the regular firing LTP, was severely impaired by the Neurexin splice site 4 in, but rescued by the Neurexin splice site 4 out. Whereas the presynaptic form of LTP was perfectly normal. Exactly the opposite as for the beta-neurexin knockout. So you have two different manipulations on the same gene family, one of which deletes a minor isoform, the beta-neurexins that are transcribed from a different promoter, and it blocked presynaptic LTP. One changes splice site 4 in one neurexin and it blocks postsynaptic LTP, demonstrating sort of the range of functions that neurexins can perform. And so what these findings illustrate as implications, first of all, for us gratifyingly, because we had put so much stake on alternative splicing, would have been kind of depressing if it turned out to be completely irrelevant. So we were happy. It had a bond. I mean, it did something. It also shows that alternative splicing, Neurexin alternative splicing, mediates synapse specification by controlling postsynaptic AMPA-receptors. You have a transsynaptic instruction in some way. The presynaptic terminal tells the postsynaptic side what to do. It also demonstrates that a presynaptic signal is required for postsynaptic LTP. It's now well established that the NMDA receptor-dependent LTP is completely postsynaptic, both in terms of how it's induced and maintained. Nevertheless, it needs permission. It's the rescue of the presynaptic side from a presynaptic signal. And finally, transsynaptic Neurexin actions actually involved signaling by binding to postsynaptic ligands, which we showed in independent experiments that I haven't time to show you. So to summarize what I try to tell you today, I try to convince you that these molecules will be informative as building blocks for trying to understand the fundamental principles that shape synapses. And I showed you that the alpha neurexins, as examined early on by constitutive knockouts, have as non-redundant function the general organization of presynaptic terminals with all parameters that we measured, in the sense that if they are gone, synaptic transmission is largely gone in slices. I also showed you that the beta-neurexins have non-redundant functions in maintaining the synaptic strength, at least at a subset of excitatory synapses, which at least in part occurs by regulating endocannabinoids, but may or likely will probably also involve other mediators as well. And finally, I showed you that yet another facet, which is alternative splicing as splicing number 4, regulates postsynaptic AMPA-receptors and NMDA receptor-dependent LTP. Now, what is remarkable, I believe, for me at least, is that all this is different. What is also remarkable is that we're only covering parts of the entire picture. We don't have a condition yet where everything is deleted, for example. We don't have a condition that examines the other splice sites. So I think that this spectrum will expand in terms of how neurexins transsynaptically and in this presynaptic terminal organize the properties of synapses. And the overall conclusion that I'm trying to get across is that various neurexin isoforms are modular effectors that specify synapse properties via competing ligand interactions. In this manner, neurexins contribute to the molecular logic that builds synapses and neural circuits. And I am indebted to many, many people. The more recent contributors to the neurexin story in my lab were Garrett Anderson, who did most of the studies on the B-Neurexins together with Katsuhiko Tabuchi, Jason Aoto, who did most of the studies of splice site 4, Stefan Maxamer, Theo Chaba did a lot of contributions on the various aspects of the neurexin story. Wei Shu did all the studies on the memory precision that I mentioned, illustrated originally. And again, Sung Jing, Jason Covey, and Özgün contributed especially to understanding how neurexins are expressed and compete with each other for ligands. I have many collaborators at Stanford without whom I wouldn't be able to do anything, let's be honest. And Rob Malenka is my neighbor and has been an active contributor and partner in all of these studies. Steve Craig, we've done some really, for me, incredibly interesting studies on gene transcription of neurexin alternative splicing. I do a lot of work on human neurons with Marius, which I haven't talked about. Axel and I perform structural studies on neurexins and their ligands, and I collaborate at all levels with Lu Chen, trying to run labs and work on plasticity in various forms. Thank you very much for your attention.
For more, please visit us at stanford.edu.